Hi, everyone. Today's webinar is titled FDA Drug Topics, Tips to Navigating Drug Interaction Information in the U.S. Prescribing Information. My name is Dr. Kara Burke, a pharmacist and team leader in the Division of Drug Information. I would like to welcome you to this educational activity sponsored by the FDA Center for Drug Evaluation and Research, Office of Communications, Division of Drug Information. We thank you and appreciate your patience as we continue to offer these CE courses completely virtual. If you experience any technical difficulties, please email us at ddiwebinars@fda.hhs.gov for direct assistance. Before I introduce our speaker, I have a few housekeeping remarks. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than only that of a single supporting company. Unapproved use disclosure. CE faculty speakers are required to disclose to the attendees when products or procedures being discussed are off-label, unlabeled, not FDA approved, and any limitations on the information that is presented. Disclosure. The faculty's planning committee members and the FDA CE consultation and accreditation team have nothing to disclose. We hope you will enjoy meeting our presenter today. Dr. Joseph Grillo is the Associate Director for Labeling and Health Communication for the Office of Clinical Pharmacology and FDA Center for Drug Evaluation and Research. The Labeling and Health Communications Group ensures labeling consistency, coordinates prescription drug labeling enhancement initiatives, and fosters outreach and stakeholder engagement for the Office of Clinical Pharmacology. Prior to this, Dr. Grillo was a senior clinical pharmacologist in the Office of Clinical Pharmacology for 7 years. Preceding his FDA career, Dr. Grillo was a faculty in the Departments of Pharmacy Practice at St. John's University and Shenandoah University, and is currently an affiliate associate professor of pharmaceutics at Virginia Commonwealth University. Now, please give a warm online welcome to Dr. Grillo.
Good afternoon, and thank you so much to the FDA Drug Topics Planning Committee for inviting me here this afternoon to speak with you about an important topic that touches both drug safety as well as health communication. Just a couple of points before we begin. Any of the tables or presentations of labeling information that I give are just illustrative examples, and they are not to be considered templates or necessarily the only way of presenting that information. There are some FDA resources that we'll talk about related to drug interaction, examples of specific drugs causing certain types of drug interactions, and these aren't meant to be comprehensive. Obviously, the drug interactions are always evolving, and these resources aren't considered comprehensive references. And these resources we're going to talk about only focus on CYP and transporter-related drug interactions. Of course, there are other types of drug interactions, such as pharmacodynamic interactions, et cetera. And then lastly, any opinions I make on my own and shouldn't be considered FDA policy. So our learning objectives, what we'll go through today is first, we'll talk a little bit about how does FDA address drug interaction information in the submissions it gets. How does it decide something is a clinically significant drug interaction? And once it does that, how does it communicate that to healthcare providers through its prescribing information? Then we'll talk a little bit about a new resource that we have specifically for healthcare providers to help with examples of some of these CYP and transporter related drug interaction classes. So for example, when you see on labeling, don't use with strong CYP3A4 inhibitors, but what would examples of those be? And that's kind of what that resource addresses. And then lastly, we'll lightly touch on an emerging technology, at least emerging in drug development, the use of physiologic-based modeling to help us identify potential drug interactions during the drug development. So drug interactions are a significant public health issue. They're a significant cause of preventable morbidity and mortality. They're responsible for a lot of hospital admissions and emergency room admissions, especially for the elderly, where they're using a lot of polypharmacy. And just given the breadth and complexity of the drug interaction space that we have right now, it's easy even for healthcare professionals to sometimes have issues with identifying these drug interactions. And one example is several years ago, the Chicago Tribune had reporters go out with fake prescriptions of very known drug interactions. And unfortunately, 52% of the time the prescriptions were filled, without warning regarding the potential drug interaction. So it is a very, significant public health issue that is preventable, provided we, know these drug interactions and, implement the prevention and mitigation strategies for them. So just a quick overview of drug interactions, so we're all on the same, page. So drug interactions, for the most part, can be broken into those that are pharmacodynamic related or pharmacokinetic related. Looking just first at the pharmacodynamic related ones on the right-hand side of the screen, they could be, synergistic. Both drugs affect the same target receptor and give a similar action. It could also be additive interactions. Two drugs that cause sedation cause much more sedation. It could be antagonistic. The subject drug affects a certain target receptor, and the interacting drug, blocks that engagement with the receptor. On the pharmacokinetic side, which is on the left side, you also have, several different types of interactions that could occur. The first would be absorption type interactions, where you could have chelation. The drug binds with food or another drug in the stomach, preventing it from being absorbed into the body. We could have pH effects. Certain drugs have very pH sensitive chemical characteristics, such that if you increase the pH with either food or drugs, that could prevent the drug from being absorbed or enhance its absorption. And then lastly, drugs actually can be metabolized and are sometimes actively transported, from the stomach, from the gut into, the systemic circulation into the body. And those can be affected by other drugs, in the gut. The other big type are binding reactions, protein binding reactions. So, drugs that are highly protein bound, let's say 98, 99%, the active moiety is really the unbound, that small fraction. So therefore, if another drug affects that binding and increases the unbound active fraction, you could have, much greater effect, much greater adverse reactions, et cetera. So to be binding. Now, by far the most common, and if you hit the next, it'll highlight, the most common type of drug interaction is the ones related to elimination and transport. And those could be related to, metabolism, the CYP enzymes, some of the phase 2 enzymes, such as the glucuronidation system. Then you have transporter-based interactions. So that could affect renal elimination, the entry of drugs into different organs, including the metabolic, organs, and, effects on excretion, renal, hepatic, and other methods of excretion. So what does the FDA look for? So what is our objective when we're looking for drug interaction? So the two big things are, one, is there a clinically significant drug interaction? And if there is, what can we do to prevent and manage it? So, how do we go about doing that? Well, so very early in development, we can tell if there's a signal. Is the drug altering other drugs, or are other drugs altering our subject drug? And that usually can be done in a laboratory model. And we could just get a signal that it inhibits this transporter, it inhibits this CYP enzyme, et cetera. That's more the PK type drug interactions. Well, but is that enough? Well, it isn't, right? So, it is enough if, in many cases, to say that there isn't going to be a likely drug interaction, but it's not enough to tell us that there is. So what's the next point? Well, the next step would be, what's the magnitude? So that's when we'll do either some type of dedicated study or modeling to quantify the magnitude of the exposure change. So it may be a twofold increase if you use these two drugs together, or this drug with this food. So then is that enough to say it's clinically significant? Well, no, it's not, right? So then we use what we call in regulatory science, the totality of evidence to really qualify the drug interactions. What does a twofold change mean for this drug? Based on what we know about the safety of the drug, the chemistry of the drug, the pharmacokinetics, pharmacodynamics of the drug, et cetera. The totality of everything we know about this drug. And then from that, we make a determination that yes, this can cause significant risk or reduced efficacy, and it is something that needs to be addressed. And then from there, we will develop a prevention and management strategy to address it. And so where does this information come from? Well, it comes from lots of different sources. So like I alluded to in the previous slide, we do have prospective clinical studies, in vitro studies, in vivo studies, sometimes human dedicated drug interaction studies. Sometimes we'll get information actually from the larger pivotal trials, the registration trials as well. We do sometimes get information from modeling. We have population-based modeling, which uses all the data that we have and sort of is able to, using advanced statistics, draw correlations between certain effects, and that could help us. And then we have other approaches, such as what we're going to talk about today, which is the physiologic-based approaches. Sometimes, once a drug is approved, as part of its life cycle, after a drug's approved, everything that happens after that, we call the drug's life cycle. We can have post-marketing information that can inform us about interactions that maybe we weren't caught in the initial development, or literature that might help with that. And then extrapolation. So a lot of times when we do these dedicated studies, we do them really with like either index drugs or drugs that we feel are very pure representatives of strong inhibition of this enzyme or whatever. And from that we extrapolate that to the class of strong inhibitors or sensitive substrates or something to that effect. And so once we decide there's a clinically significant drug interaction, here we're talking more about the pharmacokinetic drug interactions. How do we go about thinking about a management strategy? How do we come up with reduce the dose by 50% or something, something to that effect you might see in labeling? So the best way we can do that is if we have exposure response information. So if you look at this, the graphic on the right-hand side first, you could see that it looks like for ketoconazole and for diltiazem, which would probably represent the CYP3A4 inhibition on our drug, that there's potentially, there's a difference, right? Those dotted lines are the 80% and 125%, 90% confidence intervals. And we use those in regulatory science to say there's a difference. Now, whether that difference is clinically significant, we don't know yet. We just know that's different statistically. So then we go to the left-hand side here, and this is where we have an exposure response curve. This is where a PK/PD study is done, and we look at a very specific endpoint. So in this case, it's major bleeding. And we look at as the exposure, the area under the curve changes, how does the risk of bleeding changes? And we look at when someone gets no drug inhibitor or when there's a drug interaction with this inhibiting drug, which is the red line. And you can see that the change in major bleeding doubles. So assuming this drug has dose proportionality, so I double the dose, I should double the exposure, then we may recommend in this case a dose reduction. Or if it's such a serious issue, such as major bleeding, we may say, twofold is even with a dose reduction, the risk of bleeding is too much to recommend this drug in this particular scenario, drug interaction scenario, and we'll recommend avoid. So, so this is how we kind of think about these prevention strategies. So now that we have our drug interaction, we know it's clinically significant, we have an idea of how we're going to mitigate it. Now we have to communicate it to healthcare professionals, right? And the primary way FDA and drug developers, communicate with healthcare providers about all this great information. We get a ton of information from these drug development studies. How do we communicate that to the healthcare provider? We do that through our prescription drug labeling. And our regulations say that this really has to be the essential information needed for the safe and effective prescribing of the drug. So it's not meant to be a data dump. It's meant to really be the essential information to use. And you could always look at our reviews, which are available on a website called Drugs at FDA, and you could see a lot of detailed information about how we come to these conclusions. But really for the labeling, we look at essential information. The red arrows are just areas of the prescribing information sections where you might find information about drug interactions. And there's a lot of areas, but the circles are really the key areas for drug interaction information. So most of the information where you're going to find, the conclusion, what needs to be done about it, how bad it is, would be in section 7, the drug interaction section. The other section where you find more details about why that's a concern would be in section 12, the clinical pharmacology section. Will describe the magnitude of the change. And if they do recommend a dosing adjustment, you would find more details about the dosing adjustment in section 2, which would be the dosage and administration section. So generally, just a tip on navigating through the labeling: the lower the section number, the less detail you'll find in there, and the more conclusions you'll find, or more, what do I need to do about this, bottom line kind of information. And more details are in the higher numbered ones. So you have more details in section 12 about the drug interaction, and more direct information in section 7. And so starting probably in about 2015, CEDAR and as a mirror, our office, the Office of Clinical Pharmacology, began a labeling initiative with the goal of trying to enhance the clarity, utility, comprehension, and consistency of our labeling. Because the information is very highly technical and sometimes it's hard to navigate. And really, we try to look at trying to be more consistent and clear in what we're communicating to healthcare providers. And we looked at doing this in two ways. One was be addressing content. First, really making sure that we're only giving you the essential information, not writing a research report, but really giving you the essential information you need and not hiding that essential information around a lot of contextual information. The other is to reduce redundancy, not repeat information in multiple sections, use effective cross-referencing, for example, to not do that. And then the last thing, which is really important for areas like clinical pharmacology, is to minimize the use of technical language. So we have a lot of technical jargon as pharmacokineticists, clinical pharmacologists, that we would use that may not be intuitive to a healthcare professional that isn't a clinical pharmacologist, right? So we need to really address, trying to explain information rather than using a more technical term that may not be easily understood with, how do I need, it's fine that it's nonlinear, but what does that mean for me? How do I need to adjust it? What do I need to do about that? So make sure that we have that pull through. And then on the presentation side, we try to be more clear in how we're presenting information. So we use more text attributes like bulleting and white space and the like. Alternative displays, can we use a table instead of a very large paragraph? So doing that kind of thing. And just to give you an idea with cross-referencing with the example of drug interactions. So as I mentioned before, really sort of like home base for drug interactions is this Section 7 drug interactions. But, if you were to want more information about the studies that were done, the results, the actual magnitudes and stuff, you can go to Section 12, clinical pharmacology. If there's a dose change recommended, you can go to Section 2, the dosing section. If there's a serious adverse reaction or something, for example, there's a contraindication, you want to know, how bad is this? There's usually more information about that in the warning section, in the contraindication section, and sometimes even a boxed warning. So let's look first at the drug interaction section. So like I said, that's kind of home base. I tried very hard not to put too much regulatory stuff into this presentation, but just quickly, so we have some goalposts about what we have to put in there. So we have to have a description of any clinically significant drug interaction. So not everything that was studied, just the ones that we deem clinically significant, either between two drugs, over-the-counter prescription, classes of drugs, foods and drugs, so dietary supplements, other foods like grapefruit juice, for example. So a description of that would need to be in the drug interaction section. Any practical instructions for preventing or managing the drug interaction. If we know the mechanism, just sort of a brief description of the mechanism. And also in this section, some people may not know that any drug test interactions would also be put in there, like laboratory tests, that kind of thing. Some challenge we have in developing this information. Well, one of the big ones is that, especially in the area of metabolic pathways and transporters, this information is evolving really quickly. As soon as we approve, a drug, there's a couple other drugs that just have been shown to be affected by this pathway or something. So it's really a dynamic area. So it's it's really hard to be able to keep up with that, given that our labeling is not updated real time. So especially when we're approving a new drug, it may not show up in older drugs that may be involved in. So the cross-labeling thing, sometimes there's a lag in that happening, which is problematic. And sometimes we hear from healthcare providers, they have a different levels of understanding of these things and different needs. So certain specialists may want more info. So oncologists may want more in-depth information about biomarkers and whatnot, whereas generalists may just want the bottom line. So where do you find that happy medium where you're giving both groups what they need? And then lastly is there's sometimes inconsistency between the prescribing information and some of the third-party information sources that you have, because they have their own medical advisory committees, and they may decide to make a dosage recommendation where we may say we don't have enough information to recommend a dose. So there's that little bit of disconnect there. And so this is an example of what you might see nowadays in Section 7, the drug interaction section. This is a text example. And I don't know if you remember back, some of the older type labeling, but it read almost like a textbook. It was a big paragraph, and it started out with sort of, several sentences about the mechanism, the PK of the drug, and then it might get into the drug interaction, and then at the end, it might get into what you should do about it, but it's very hard to navigate. And what we heard from healthcare professionals when we asked them is, we kind of want the bottom line. The three things that need to be super clear in the label is there's a problem, there's a drug interaction, how bad is the drug interaction, and what do I need to do about it? And while sometimes we would like to see some of the context as well, those 3 points need to be super clear. So we heard that, and based on that, we've kind of revised how we do our labeling. And you can see in this example, right up front, the main action is communicated. So if you just look at the first couple of words, these 2 drugs, I need to make a dosage modification, right? And then if you want to know why, you can go further down, and it gives you sort of a description of what the issue is, what the mechanism is. And if you want even more information, it gives you some cross references to different sections where there is that additional level of detail. Now, the text format is okay when you have 1 or 2 drug interactions, but when you have multiple drug interactions, then you may want to consider using more of a table format, like 3, 4, 5 or more drug interactions. So, and this is just one way that we've recommended that you could use a table. And this just gives the mechanism, it gives the prevention and management strategy, and you could flip those around and put the prevention and management strategy first, so it's right up front as well. Another time where you would want to use this, even if you don't have a lot of drug interactions, is if it's a specifically complicated drug interaction scenario, such as the one here with the gastric acid reducing agents, where you have multiple prevention and management strategies. Putting that into text can sometimes be a little difficult to read, navigate, and so a table may be better in that case as well. We even have another table, which I haven't shown here, but for like the antivirals, where they have a ton of drug interactions, where it really is able to accommodate that amount of drug interactions, but you don't get as much information, unfortunately. Just one area that you might see, and this is sort of semi-standard language. So when you have therapeutic proteins that are indicated for diseases that cause inflammation, one thing we know is that the inflammatory process affects the cytokines, and those could suppress CYP enzymes. And so when you give a drug that helps the disease, right? It improves the disease, reduces the inflammation. Over time, those CYPs are going to, come back up again, and you could find drug interaction. You may need to make dose modifications to some of the substrates of those drugs. So this is a warning that you might see for some therapeutic proteins, saying as you're treating this disease, you may see a change in some of these other drugs because the metabolic pathway the metabolism is increasing due to removing the suppressive effects of these cytokines. So the FDA has several resources available for helping different groups with drug interactions. The one we're going to focus on today is this new one that came out this past fall for healthcare professionals. It's a site that gives examples of drugs that interact with CYP enzymes and transport systems, so that, for example, in the label where you see do not use with strong CYP3A4 inhibitors, you could go to the site and see, some examples of, what those might be. Again, not a comprehensive list, but a list that was evaluated by FDA and that we felt good that we could add it to the list. Another is a public site. It's actually being redeveloped right now, and we're hoping to have that up hopefully in the next few months or so. And that's going to be in a very less technical language, trying to explain to lay people different drug interactions, and it'll hopefully have some case-based presentations as well to give them a feel for what these different types of drug interactions could be and why they need to talk to their healthcare providers when they're changing their drugs or using OTCs or changing their diet, etc. And then the last one is the site for industry, and that is in a lot of ways similar to the healthcare provider table, to the healthcare provider site, but it's really just a series of tables. So while it's okay for folks who are developing studies for drug interactions, it's really hard. It's not as user friendly for a healthcare provider, and that's why we developed this new site. And this is a screenshot. The little blue box there is a screenshot of what the site looks like. And really the benefit of this site compared to at least the industry site that we have, is this gives you a complete picture of a drug's metabolic and transporter based, profile based on, the information in the database, obviously. So for example, if you're just looking in this example, you're looking for, 2CH strong inhibitors, right? And you'll find gemfibrozole, but it'll also tell you that gemfibrozole has some transporter issues as well. So you're not having to search through multiple tables, a table of transporters, a table of this. It all gives you all that information in one, one result. And what's on the lower side of the lower end of the slide here is what you would get, the results you would get if you put in that CYP2C8 filter. And so how to use it? Well, there's several different ways you could look for information. One is drop-down filters. And if you look a little bit to the left there, you can see what one of those drop-down filters would look like this is for a CYP example. It's different for inducers, inhibitors, sensitive and moderately sensitive substrates, and different transport inhibitors and substrates. You could do free-form searches, and you could also export your findings, or if you leave all of the sections blank and hit export, you could export the entire database itself in Excel format. Some tips for navigating. One, we spoke about the filters. One important thing to remember is, it's searches based on the drug substance name. So rather than the proprietary name or the salt, the salt name. So you see those examples. It ignores certain punctuality. So for example, with the St. John's wort, if you leave off the period after Saint, you're still going to get the results. And remember that when you put, especially in the search box, two terms, it's an and function. There's no or function. So you'll be getting the findings for both situations. One caveat is if you're looking at this with a small screen, for example, a phone. When you do that, if you look on the left-hand side of the screen, you'll see, this is kind of a truncated result, right? Where's my transporters? Where's my substrates? Well, they're actually there. But what happens is if you have a small screen, that little blue plus sign will show up. And if you click that, it'll drop down and give you those other missing columns. So just remember that if you're not seeing the complete results, inhibitors, inducers, transporter inducers, inhibitors, substrates, etc., look for that blue plus sign and it will give you all the information. So let's move on to the clinical pharmacology section, which is where all the detailed information is. Mostly, and we'll look really at the PK section, which is subsection 12.3. I'm not going to go read through all of this stuff. I just want to highlight just some of the blue information, that the information you'll find relative to a drug interaction will be information about the drug's metabolism, transport, which could be important for drug interactions. You'll find the results of the dedicated in vitro and in vivo, so laboratory as well as human studies done specifically for drug interactions. But you won't find any of the recommendations and stuff. That's really not supposed to be repeated in this section. That's in section 7. And this is just an example of the drug interaction information you would find in 12.3 as text. As you can see, again, it's really just the essential information. We're not writing a research report here, so we don't give, exhaustive details about the study itself unless it's clinically important. So if it's important, like for example, the dose of inhibitor used is, lower than that used clinically, and you should know that these results, you might see a greater change in exposure than shown here, because in practice, you'd be using a higher dose of inhibitor. That information would definitely be in here. But you can see it's a very straightforward. It gives the expected change in exposure. It gives some of the basic conditions of the study so that, you can discern, the information, critically evaluate it as you need to. The other thing that we give is the in vitro information. And, this is sort of a mixed bag. Folks say, well, what's the use of it? Because we have the clinical studies and those really inform the clinical significance more. And really, the way I look at it, and again, this is personal opinion, not FDA policy, is I think it's important for a healthcare professional, in case you're looking in section 7, you don't see something you're thinking might be there, could be there, and you say to yourself, well, did they study this, or could this be happening and they just haven't studied it? So you can go here and say, well, okay, they did look at it. It's not clinically, wasn't significant in vitro. Now, obviously, it could be that this is a red herring and there is a drug interaction that we didn't pick up or something, but this is just a way so you can have a complete picture of what we looked at, or what was looked at by the drug developer with regard to drug interactions. And again, depending on the breadth of information you have, again, like the antivirals, for example, have a lot of drug interactions. So to put it in text would be very difficult to navigate for the healthcare provider. So in those cases, we can use tabular format, or sometimes they could use a figure. I'm not as big a fan of the figure, especially you'll find in the older labels, because you'll see. So if you look at this, right, again, we had that 80 to 125, 95%, 90% confidence interval. You'll see, what's outside of that range. So you would say to yourself, well, these must be significant drug interactions, but actually they're not. It's that situation where, based on the exposure response, even though they're different, they're not going to cause a clinically significant risk or reduction in efficacy or something like that. In this case, it would be risk. So you have to be very careful with these, because really the clinically significant drug interactions are in section 7. Everything that was kind of studied is in section 12. So, you'd have to be able to discern that. So we're going to get into the physiologic-based modeling, which, again, I say is an emerging technology in drug development, but believe it or not, it's been around. The earliest study was in 1924, looking at inhaled ether. So it's a technology that's been around, used a lot in toxicology, more recently came into the drug development field. And I just want to get just an idea of, how many healthcare providers out there have heard of this term physiologic-based modeling, in your clinical practice environment, either studies you've read or in labeling that you've looked at or something. If you could answer that survey, I'll just wait a couple of minutes. I would appreciate it. And I know the industry people know it, so you don't have to answer. Okay, so that's actually pretty good. So we have 60% said no, but I'm very happy to hear 42% said yes. So that's really good. Do you feel comfortable critically evaluating PBPK-based information you see in the prescribing information in your clinical practice environment? Again, yes or no. And this is, again, more geared toward the healthcare professionals in the audience. Well, almost the same. So that's very encouraging. Great. Okay, so physiologic-based modeling is, as I mentioned earlier, something it's an old technology that's found a new use in drug development. And I wanted to touch on, and this is a very light touch, so this is going to be a heavy modeling type discussion, but just to give you a flavor for how it's being used and things you need to think about as healthcare providers. And if you want more information, like if you want a dedicated, presentation just on this type of modeling, you can give us some feedback on that, and we'll try to make that happen. But the unique thing about physiologic modeling, and why I like it as a healthcare professional myself, as a pharmacist, is that it's very clinically intuitive. Its basis is physiology. So unlike the population models, where when I'm telling you the results of the model and how the model was designed, a lot of it is advanced physics. When I'm telling you how a physiologic-based model is designed, it's all physiology. So we have parameters for blood flow, we have parameters for organ size, we have parameters for drug absorption and different drug-specific parameter effects. We have parameters for the chemistry of the drug, how much it likes to get into oily things, and which would mean it may more likely want to get into certain types of organs versus others. So it's all really a clinically intuitive way of modeling a drug, how a drug moves in the body and is eliminated. So it's actually something that healthcare providers could really dive into, really understand and help folks perhaps in their institutions develop these models, because this is where you guys shine. I mean, this is your thing. And the approach that we use is predict, learn, confirm. So this is what we call like a bottom-up approach, where the population models are bottom down. Bottom down means they have a lot of data and they sort of filter it out. Here we build this model and we make these predictions, and as we get more data, we refine our parameters and get the model more dialed in. And this is just a quick look at, just to show you the scope of what we're seeing. So, we started getting applications in 2008. I was actually involved in this first application that came in. It was an IND. Up until now, to where you can see, there's been, quite an increase in the number of submissions, total submissions that we're getting. And then if you just look at, numbers, you'll see that we have in the past, several years, a fairly consistent number of these modeling approaches coming in regulatory submissions. Those would be the green lines, the NDAs, and the gray, the BLAs. And what's also very interesting is the orange line, which is the IND. So these are drugs that haven't, been submitted yet and are in the pipeline stage. So this is sort of the future. So in the future, we're planning to see probably even more of these modeling type things, in the drug development PK. And this is the clin pharm space. Where are we seeing the most? As you imagine, it's more in the more vulnerable populations. We have a harder time doing studies, so a lot of oncology, neurology type applications we're seeing. And so I have a couple of examples that follow. I'm not going to dive very deep again into these. This isn't a modeling talk, but I wanted to show you, how we're using these models. So one way is, when we do these drug interaction studies, the approach in drug development is very binary, right? We want to take a very, clean population of healthy volunteers with very specific inclusion and exclusion criteria, and really study a very, drug very well known to have this either, inhibition or induction or whatever, and really just study it in a very clean binary fashion. But that's not the real world, right? In the real world, you have triple drug interactions, you have drug disease interactions, all these complex scenarios that often aren't captured in drug development. Well, physiologic based modeling can handle those scenarios. And this is actually an example of a study I did with colleagues in OCP. And what we had here was, this is rivaroxaban. Obviously, it's a factor 10 inhibitor, anticoagulant. And at the time, its indication was for hip and knee replacement. This is one of the earlier indications. Has much more indications now. And we had multiple studies, right? But we had these studies that were in mild and moderate renal impairment that really didn't show. It showed a difference, but not one that we would consider clinically significant. And then mild and moderate CYP3A4 inhibitors, we didn't really see a clinically significant difference, but we saw that there was an effect. And we said, well, this is going to be an elderly population that very likely has some renal impairment and probably might be on CYP3A4 inhibitors, such as amiodarone or something like that. So maybe it would be worthwhile for us to look at this and see, is there a signal? So we developed this model, and just to give you a schematic of what that looks like, that's that black and white picture there on the left. And in doing that, we were able to see that when you combined the renal impairment with the mild to moderate inhibitor, that did cause possibly a clinically relevant drug interaction. And we were able to put into the labeling, use only if the risk justifies the benefit. And so that was one application to help predict complex scenarios, which are more real world. And again, to bring it back to the clinical setting, as pharmacists, physicians, et cetera, you could do these studies too and look how these complex scenarios in your particular area can affect drug kinetics. All of these, they're commercially available packages to do this kind of thing, and they have classes to learn and whatnot. Another example would be to reduce clinical trial burden. So obviously, more clinical trials we do, the more patients are potentially or healthy volunteers are exposed to an investigational drug, increases the cost of drug development, which could be passed on to patients. So if there's a way that we could safely, minimize the number of actual clinical studies we need to do, that would be a benefit. So this is an example of where we're able to do the worst case scenario as clinical studies. So strong inhibitor, ketoconazole, strong CYP3A4 inducer, rifampin, and we got that information. So we know what the worst case could be for CYP3A4 drug interactions with this drug, right? And then from there we said, well, Yeah, and this isn't a study we did. This is the sponsor did this study. And first they said, well, can we verify the model? Can we predict, can we predict what was shown clinically? And that's those green hashed boxes. And once you're able to do that, then use the model to predict moderate and weak inhibitors and moderate inducers. So that saved 3 or 4 studies that would have had to been done and save human study exposure in these people. And just taking it one step further, so it's along the same lines of reducing trial burden, it's hard to do in certain populations. So for example, you don't want to give you can't give healthy volunteers certain oncology drugs, right? Because it's chemotherapy, it's toxic, so we can't give healthy volunteers toxic drugs. So you have to give those to cancer patients, and then doing that, they're not that clean population. They have other morbidities that could influence the pharmacokinetics, so you don't have that clean information. So if we can use this kind of modeling in these more quote unquote vulnerable populations, oncology, pediatrics, pregnancy, lactation, et cetera. And we're not there yet, but some of these we're starting. It would be a great benefit because we can get this information in these populations without having to subject children to clinical drug interaction studies or what have you. And this is an example of that, where we were able to characterize a drug interaction in a child and adolescent population from adult information that we had. And what they did was, first they took the adult information and the adult drug interaction information. They modeled, can we model the adult information using, can we effectively verify the model against adult data? Can we verify the interaction against the adult data? Then can we, create a pediatric model? And then from that, can we create a drug interaction model? And so that was done, and that was actually included in the labeling for pediatric studies. So it saved doing these studies in children, which can be a challenge. I mean, obviously, those who are parents out there, putting your child into a clinical study for drug interactions versus therapy can be challenging. And just again, we had given, and we actually did a lot better actually than this survey. We had done a survey several years ago of physicians. It was generalists and specialists, about 50-50, and we had asked them about modeling and simulation, and 25% of them needed us to tell them what modeling and simulation was so they could answer the question. And I just bring this up in that there is definitely a need in education, curriculums and continuing education for healthcare providers, to learn about this modeling, because it's increasing. It's in labeling now. So the expectation really is that you'd be able to discern, critically evaluate this information in a similar manner than that you would be able to clinical study-based information. And if you can't, then we need to get that information to you, either through educational programs or whatever. But it's important. It's something that's really the future. So if I were a student now, I would make sure I knew about this stuff as a pharmacy student today. This is the last slide. So just some parting thoughts. So again, drug interactions are a major contributor to morbidity and mortality. So anything we can do to accurately characterize them and have them have good risk mitigation strategies and whatnot would be beneficial. The FDA's primary way and the drug manufacturer's primary way of communicating this information to you is through the prescribing information, and we went through the different areas where how you can navigate that. We do have that new online resource. I hope you do take a look at it. If you have any feedback, please, we gave you an email that you could send it to. I'd love to hear it. It will get open because I will be the one who reads it, so I can guarantee you that it will be looked at. And lastly, again, the modeling is becoming increasingly used in both drug development, especially in the area of drug interactions. So it would definitely be in your best interest or in the interest of to become more aware of this technology, and for educational programs to start incorporating it into their curriculums to whatever extent. And again, clinicians could be extremely helpful to the modelers who are developing these physiologic-based modeling, because you guys really are the physiology experts. You know your diseases, you know how those diseases affect the body.
Thank you, Dr. Grillo. That was a great presentation. And that concludes the presentation portion of our webinar.
So I see one question asking about, are there any, I'm assuming it's open source resources for modeling. There's one program, I believe it's called PKSim, that's open source for doing the PBPK modeling. And you could also develop your own. I mean, one of my fellowship projects was to do a PBPK model, and I had to do it, myself with back then a 486 computer. So yeah, you could even build these models yourself. But the commercial software is, or the open source software is nice because it has some of the parameters already built into it. But there are open source, as well as the commercial companies, I believe, also have special academic rates, very inexpensive for teaching purposes and stuff. So you could look into those as well. Another question: how often is the healthcare provider website updated? Unless, obviously, if there's a very significant issue that needs to be addressed immediately, we would do that. But the plan is that we update it quarterly. Have I experienced the use of artificial intelligence? Personally, I have not, but I know that there's a lot of work being done in that space. So I guess, the future will tell us, what with that. I have not done any of that myself. There is some talk about using artificial intelligence to inform some of these physiologic kind of models. But again, that's future kind of thing. That's like, in its infancy. There's one question about when should you use the table format? And so FDA doesn't have a requirement for when you use it. What we have in our guidance document to industry is, the data, the information should be presented in the format that best suits the readability and comprehension. So, and that's based on the breadth of the information, the complexity, so we're not really, so we haven't like pigeonholed, if you have 5 interactions, you must use a table kind of a thing. So it's really, because you could have one interaction so complicated that a table or some other way of bullets or something may be needed, so we don't really, push them, we don't really hold them to a specific, format, but what we try to tell them is, obviously if you have, a few, like, rule of thumb that, again, this is my personal opinion, not policy, the 3, 4, 5 drug interactions, you may start thinking about a table might be a better way to present it than text. So there's no hard, fast requirement for when you use a table, other than, the breadth of information, the labeling has to be readable. So if we get it and we see that, like, if my first thing is, if I can't understand the label when I first look at it, then, then I'll recommend a table or something to the applicant.
Dr. Grillo, thank you so much.
Thank you. Thank you everyone for your kind attention.
Absolutely. If you have questions that we did not have time to address today, please email them to our Division of Drug Information email address at druginfo@fda.hhs.gov for assistance. If you need more CEs for your license renewals this year, please check out our list of home study CE webinars at www.fda.gov/DDI webinars. And if you have your phones out, which I'm sure you do, we have 6 QR codes on the slide in front of you that are easy to access. There's a QR code for X, the Drug Information Listserv, our continuing education webpage, our podcast, our Gaddis LinkedIn group, and our fellowship program. Please scan these QR codes for more information and CE opportunities. You may also find additional free CE credits, such as FDA's drug regulation course titled FDA's Role in Public Health, Drug Efficacy, Safety, Quality, and Beyond on our Cedar Learn website by entering CDER Learn in the search engine on fda.gov. This concludes our activity for today. The FDA appreciates your participation in this webinar, and we hope that you will join us again in the future. Thank you all for attending, and please stay safe and healthy. Take care, everyone.